So something that is very commonly confused when we're talking about chromosomes, pairs of chromosomes, homologous pairs. Sometimes we talk about sister chromatids in a pair to make one dyad. Two dyads that are the same kind make a homologous pair, or it could be two monads that are the same kind. And once we work through those concepts, then there's still some confusion about what we call the ploidy of the cell. So the term dyad and monad and sister chromatids and homologous chromosomes, these are all words referring to the chromosomes, describing the chromosomes. They are adjectives for the chromosomes. But the word ploidy, like diploid, haploid, and you can even have triploid, tetraploid, that refers to the content of the cell. So ploidy is a descriptor of the cell as a whole. And so when you say a diploid cell, you never say a diploid chromosome. That doesn't make sense. You can have a dyad chromosome. You can have a replicated chromosome, but you can't have a diploid chromosome. That doesn't make sense. Diploid refers to the cell. And so the cell's ploidy refers to how many sets of chromosomes it contains. And that word sets is confusing because it's not the same as pairs. So a human cell would have 23 pairs, and each pair means 2. So 23 pairs is 46 total chromosomes. But that is 2 sets in humans. So what I did was I created this little visual aid here. When a cell is in metaphase, that's when the chromosomes are in that replicated form, that dyad form. And under the microscope, you can get the cells to burst open and the chromosomes scatter around, and you photograph it, and then digitally you cut out each chromosome, and you have to have a lot of training, and you can figure out which 2 of these chromosomes are chromosome 1, which 2 of these are chromosome 2, which 2 of these represent chromosome 3, and so on. And that's called cytogenetics. So I've worked with some cytogeneticists, although I am not trained specifically to be able to do this, at least not as quickly as they can. But this shows, this image, I know it's a black background, but this image shows after they have digitally cut out the chromosomes from the microscope image and arranged them, this is what you might see. So 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, and so on. Because it's metaphase, when they take the photograph originally, the chromosomes are always going to be in the dyad form. And in this picture, you can actually make out the 2 sister chromatids in the dyad. Sometimes it's too blurry, and all you really see is like one solid bar for this copy of chromosome 1 and one kind of solid bar for the other copy of chromosome 1. But if you have good resolution, good clarity, you can actually make out the 2 sister chromatids for each of those copies of chromosome 1. And so you have 2 of each, and this shows the arrangement. This is arranged correctly. And then at the end, chromosome 23, so to speak, is the X chromosome. So this would be in a cell. These chromosomes were photographed from a cell that was a female. In any case, so that shows 2 sets, but it's hard to see the 2 sets. One set would be 1 copy of chromosome 1, 1 copy of chromosome 2, 1 copy of chromosome 3, 1 copy of chromosome 4, etc. And the second set would be made up of the other copy of chromosome 1 and the other copy of chromosome 2 and the other copy of chromosome 3, etc. It's easiest to visualize the concept of sets in meiosis 1, metaphase 1. And so what I did was I kind of cut and pasted from the previous picture, which, by the way, I borrowed from an open source textbook, all these images. But what I've shown you is how they would line up in metaphase 1 of meiosis. So here at the top you can see the 2 copies of chromosome 1, the 2 copies of chromosome 2, the 2 copies of chromosome 3, and then I got tired. Pretend like I did the same thing for chromosomes 4 through 20, and then chromosome 21 down here, chromosome 22, and then the 23rd pair is the X chromosome here. So what you can see is if I draw a line, which I don't have a pen on this software, but if I drew a line right through the middle of each homologous pair, so a vertical line cutting this in half, you would see that 1 copy of chromosome 1 eventually is going to move to the left and 1 is going to move to the right. 1 copy of chromosome 2 will move to the left and the other one will move to the right, and so forth. So 1 full set of chromosomes will move to the left and 1 full set of chromosomes will move to the right. And so that's the separation of the 2 sets. So each daughter cell, each product cell at the end of meiosis 1 has 1 full set of chromosomes, 1 copy of each different size or number of chromosome. Now I realize that those chromosomes are in the dyad form, but remember, when they're in the dyad form, we only count them, they're referred to as being 1 chromosome. However, in meiosis 2, the dyads will be pulled apart, and then you would get 2 monad chromosomes from each dyad chromosome, ultimately in the final products of meiosis. Each gamete will have 1 copy of each chromosome type, 1 full set of chromosomes, and they will be in the monad form. All right, so the first division, the first meiotic division, just pulls the 2 dyad copies of each chromosome type apart. And then in the second meiotic division, the sister chromatids are pulled apart. So I hope this helps in terms of sets. So human chromosomes have, a diploid cell always has 2 sets, but in humans that's 23 chromosomes in each set. In mice, it's 20 chromosomes in each set. In fruit flies, there's 4 chromosomes in each set. But still, a diploid cell would have 2 full sets. So for fruit flies, each set has 4 chromosomes, so a diploid cell would have 2 sets, which would be a total of 8 chromosomes. So the number of chromosomes in a set is specific to any species.